Ok, lo streaming sta partendo. Va bene, cominciamo la lezione. Se c'è qualcuno che ha delle domande o dei dubbi, in questo momento, se volete, io aspetto un attimo e se mi ponete le domande io vi rispondo, se no fra un minuto cominciamo. Ecco, vedete lo schermo. So we started by introducing general concepts that you may employ for building an ecological model of a living system or an ecosystem. It is quite normal to start asking ourselves, what is life? This is why I titled the lecture of today, What is Life? The second title of the lecture is Gaia's Epigenetics. So we will discuss, according to what we have introduced on Monday, how we can define living systems from an operational point of view. What is life? I anticipate it now. It is the title of a very well-known booklet written by Erwin Schrödinger in 1944. Erwin Schrödinger, you may recall, it is a very famous physicist, one of the founders of quantum mechanics, but in the late part of his life started to ask himself this question, what is life, and try to find some answers. As we will see, these are quite an intriguing questions because it appears as if living systems are somehow violating some of the most important principles of physics, like, for example, the second principle of thermodynamics. We'll discuss a little bit more about that. Let's start back by recalling that the energetic base on which all living systems are based on our Earth planet comes from the sun, so our main star. And we said that we may measure the quantity of radiation coming from the sun by employing the solar constant, that is 1,340 watts per square meters at the intermediate distance Sun-Earth. If we make 100% this quantity, we have said that 35% of its energy is immediately reflected by clouds, dust, gas, and albedo. 90% is absorbed by the atmosphere, and 46% is absorbed directly or indirectly by the Earth itself. We said of this quantity of energy, only 5% is the one that is responsible for all atmospheric circulation, accounting for the heat of evaporation and convection. We said also that our planet seems to be in equilibrium from an energetical point of view, in the sense that what it is absorbed coming from the sun, it is exactly re-emitted. So that if you make 100% minus 35%, you obtain exactly 65%. So the equilibrium is obtained. So what it is arriving on the surface of the planet, it is also what it is remitted from the planet. We started by giving a look to the energetic efficiency of transfers on the planet. We said that we may convert the solar constant 1,300 and something watt per square meters in kilocalories per year directly incident on the surface. So this figure amounts for 1.2 10 to the 22 kilocalories per year. This energy is what it is employed for sustaining primary production. Primary production, from an ecological point of view, it is referred to as terrestrial vegetation and phytoplankton. Please note one of the most important primary producer in our planet is phytoplankton, so small algae in the oceans. Of these 1.2 10 to the 22 kilocalories, primary production employs 2.5 10 to the 20 kilocalories per year to survive, and by employing this energy, primary production produces 5.2 kilocalories per year of biomass. The efficiency of this energy transfer is quite low, 0.2%. But then we said that going to the upper trophic levels, the efficiency tends to grow. So the idea is that with an decreasing efficiency, with an increasing efficiency, there is energy conversion factors. We said that primary production from a process point of view is called photosynthesis. So synthesis of organic molecule. This synthesis is made by employing solar energy, as we have said before. The reverse process of primary production is called consumption. Consumption, that biologists or ecologists may call fermentation of respiration. Consumption means conversion of organic molecules into ATP. ATP is the energy fuel for all cellular processes. ATP is a molecule that transports energy into living cells. We said that from an energetic accounting, we may convert mass into energies. These are the classical factors of conversions. So you may say that plants have a specific energy content that is lower than algae, that in its turn is lower than invertebrate, when it is then lower than vertebrate. So we may say that different types of organic molecules have a different content in energy. We also said that these factors may be converted in terms of different types of organic molecules. For example, carbohydrates, protein, and lipids have all a different content in energy. And so the different living objects like plants, algae, invertebrates, and vertebrates are made up by a mix in different proportions of these elementary organic molecules like carbohydrates, proteins, and lipids. So but the number we may employ for the conversion between energy and mass are this one, exactly. Then we turned ourselves to asking more in detail what is life, where life comes from. In order to answer to this question, we made a very quick history of the biosphere. We said that we may subdivide the history of the biosphere in three macro ages: prebiotic, microcosm, and macrocosms. Prebiotic, so before life appeared. Life appeared for 3.5 billion years ago. So the prebiotic age goes from the burn of our planet, that is 4.5 billion years ago, up to the appearance of living system, 3.5. Then we have a microcosms, that is a very long period in which microbacteria or microorganisms dominated life on our planet, and this goes from 3.5 to 1.5 billion years ago, so for a time span of 2 billion years, bacteria dominated life on our planet. And then we have a macrocosms that started 1.2 billion years ago, in which all large living organisms appeared on the planet. We have made, we said, first of all, that bacteria that have dominated the microcosm, so a long period of the story of evolution of our planet, have changed in a dramatic or significant way the condition of our planet. In particular, they adjusted the levels of various elements in the atmosphere, for example, humidity, hydrogen, CO2, oxygen, and ozone. We have made a projection of the history of evolution on 24 hours. So we subdivided it into time spans, night and day, and we said that large organisms appear very late in this projection. So we say larger organisms appear in the evening of the day after the appearance of living objects. It is interesting to see what life has done to our planets. I recall you that we are trying to answer what is life. So it is interesting to see and to understand what life has done to our planets, for example, by comparing some characteristics of our planets in our solar system. For example, if we look at Venus or Mars, and we compare it with the actual condition on Earth, we see that we have a very low concentration of CO2 in the atmosphere compared to what is happening to Mars or Venus. And we have seen that by modeling approach, this figure, 98.0%, will be the content of CO2 if life was not present on our planet. So it is quite apparent that the presence of life on our planet lowers the concentration of CO2, lowers the concentration of gaseous nitrogen, lowered the concentration, or enhances the concentration of gaseous nitrogen, enhances the concentration of oxygen up to 21%, and similarly, other particular characteristics are regulated by the presence of life. For example, we have a 14 Celsius degree average temperature on our planet, but compared to the condition on other planets, it is a suitable temperature for living systems, for life to thrive, while on the other planets, the conditions are very, very different. For example, very high temperature on Venus and very low temperature on Mars, but is much farther away. Again, for the pressure, we have the same conditions. So the atmospheric regulation effect of living system has brought a pressure of 1 atmosphere on our planet, while pressure are significantly different on other planets or on our condition. For example, on our planet, if life was not there, we might have much larger pressure. Like, for example, on Mars, we have a very, very low atmospheric pressure. So you see, it is quite evident that somehow life has changed the condition on our planet. So let's stress again the key question: what is life? That is the title of a booklet of Erwin Schrödinger, 1944. I suggest you to read this booklet. There is a very nice edition of Adelphi. This is a 80 pages booklet, very, very interesting, in which Schrödinger tried to give an answer to these questions, looking at it from the physicist point of view. It appears apparent to a physicist that living object seems to defy the second principle of thermodynamics. I recall you that the second principle of thermodynamics tells us that disorder or entropy is always increasing. All thermodynamic processes, according to the second principle of thermodynamics, entrains the fact that the entropy of the system is growing. We have seen, or we have said, that entropy somehow is a measure of order, or negative entropy is a measure of disorder. So, saying that living object seems to defy the second principle of thermodynamics means that if we look at what happens with living things, we may say that the idea of that living things tend to increase the disorder of the system is somehow contrasted by the evidence or the apparent. For example, I told you, if you look at a tree is a complex structure in which there are some kind of shapes or patterns that are repeated all over the history of evolution of a living object. We may make the same observation by looking at animals, for example. When we look at animals, we find hard to conclude that a living object tends to go to states of increasing entropy or increasing disorder. So what happens in reality is that we observe that life or living objects is locally decreasing the level of disorder, or is locally decreasing entropy. I anticipate immediately that the second principle of thermodynamics is anyhow saved by the fact that if we make the budget in entropy, including the whole system, so not only the system at focus, but also the surrounding environment or the surrounding system, we discover that if we make the summation of entropy changes into living system, and we sum it with entropy changes into the surrounding environment surrounding to living system, we found that there is always an increase in entropy. So the entropy of a whole system, or the system looked as a whole, is increasing, but what we may say is that at least locally, life or living systems seems to decrease entropy. So the answer to the question, what is life, starts from recognizing that living objects are objects that at least locally tend to decrease the entropy, while if you make the calculation of the whole system, the entropy is always increasing. Do you have any questions about that? Is it clear? Okay.
Yes, it's clear.
Okay, thank you. It is important to notice that Schrödinger, in his booklet, What is Life, made two instances, very important instances. The first important instance, what he has called order from disorder. It is a very impressive anticipation of the theory of far from equilibrium thermodynamic system, in which it was apparent that, or it appeared quite clear that systems, thermodynamic system, far from equilibrium, not too far from equilibrium, but not in equilibrium, thermodynamic system tend to onset or to manifest dissipative structures. Dissipative structures are structures that convert forms of energy, for example, mechanical energy, into other types of energy, for example, thermal energy. A dissipative process, as you may recall, it is a process of friction, for example, in which you have that the movement of an object is converted into heat. This happening, this story is happening ubiquitously, so everywhere when you look at something more than simple, so a little bit, at least a little bit complex thermodynamic systems in which you see the appearance of dissipative structures, but seem somehow to contrast the tendency to increase in entropy. There is an interesting example of this, the so-called dissipative cells of Benard, in which physicists have discovered that if you put a liquid between two metal plates and you impose a temperature gradient between the metal plates, so for example, you heat the lower metal plates and you leave the upper metal plates to be cold, after a certain critical difference in temperature, if we call delta T the difference in temperature of the two plates, after this difference in temperature increases over a critical value, we may call delta T critical, what happens is that there are some kind of convective cells that appears. Let me see, I will try to draw something, please forgive me, because my hands are a little bit not very controllable. So let's see if I can write something. Okay, we have two plates, two metal plates, and in between the two metal plates, we may see that there is some liquid. We show the two metal plates, so that we show the lower one, so that we have a temperature one that is increasing compared to a temperature 2. So the upper plate is colder than the lower plate we are showing. What it happens is that after the delta temperature grows over a certain critical limit, there is a dramatic changement in the structure of the system, and these dramatic changes, delta T critical, and this changement is manifested by the appearance of convective cells. So inside this liquid between the two plates, there are kind of zeros, or how do you say, cycles, convection cells. Of course, convection cells maybe from one direction. This is a clockwise conversion cells. Of course, the parallel one becomes a counterclockwise convection cells, so that you may immediately understand that the space or the liquid, after this growing of a difference in temperature, becomes somehow informed. Some information becomes substructured. For example, I can distinguish this part from this part by looking in which direction the liquid is flowing into the convection cell. For example, this is a right convection cells, and on the contrary, this is a leftish convection cell. So we have a distinction of the space due to the fact that we have an onset of convective process, and this onset of convective process is driven by an increase in the difference in temperature. DNA. So this is the first instance of Erwin Schrödinger order from disorder. So even in a disordered system, if some kind of dissipative processes start up, then some kind of ordered structure appears. I'm not talking of ghosts, or I'm not talking of fake news, or I don't know. I'm talking about things, but of things that really happen in reality. I'm saying that if you put a liquid between two metal plates and you impose a gradient in temperature between these two metal plates, it is an observation that there is the onset of convective cells. And this was so striking, so impressive, that gave rise to a particular research subject in physics, in particular in thermodynamics, the so-called thermodynamics of far from equilibrium system, that was developed by Prigogine of the school of Brussels in the 60s of the 20th century. And they have been able to demonstrate that, at least locally, in certain conditions, the entropy of the system may decrease by the appearance of this kind of structures. Then the second instance of Schrödinger was called order from order, and this is even more impressive because Schrödinger recognized that somehow the appearance and the development of life should be linked to the appearance of some kind of ordered or informed structures, some structure that contain some information concerning the living system. I recall you this booklet is 1944, and I anticipate, or I recall you that in 1954, there was the discovering of DNA by Watson and Crick. So 10 years after the anticipation of Schrödinger, this instance order from order was being manifested by the discovery of DNA, and DNA, it seems to be exactly the place in which, of the molecular biochemical structures in which the information related to the construction or to the structure of a living object is contained. All living objects contain DNA. DNA is the first molecule that is duplicated and transferred in a cellular replication, for example. And it was quite impressive that 10 years before the discovering of this molecule, these physicists, Erwin Schrödinger, have the intuition that there should be somewhere in living system a place in which storing information concerning living objects. I mean, nowadays this is quite evident because we are used to think or to reason in these terms, but believe me that in the 40s and in the 50s it was not so evident to imagine this. Even in 54, when DNA was discovered, it took some decades in order to understand exactly the structure of DNA and in order to understand exactly its role in biochemical processes and so on. Then Schrödinger, in the booklet of 94, also introduced some concepts that were easily inherited by physics, in particular by statistical mechanics. Schrödinger suggested that there should have been some kind of law of a square of n. So the square n is the number of events. So the idea of this order from order and order from disorder, the linkage between these two instances was to be found in the fact that statistically speaking, the number of mechanical processes that are involved in the changement of the structure of DNA is linked to the large number of events, colliding events. I'm not going too much into these statistical reasonings, but just remember that the inaccuracy related to our understanding or knowledge of a process decrease or tends to a static or tends to an asymptotic value as the square roots of n. This also explains and was made quite clear by Schrödinger in his booklet that the changement of manifestation of life was linked to some kind of changement that was called like mutations by Darwin in the end of the 19th century. So mutations are induced by the external environment. Maybe, for example, from the point of view of a physicist, it was quite normal to imagine that X radiation was that responsible of causing mutations, in particular in the process of DNA replication, and this gave rise to the whole variability of living objects as we know them today. Okay, am I being too exotic, or you can follow what I'm saying? Okay, now let's try to understand more specifically how life appeared on our planet. We said that our planet is 4.5 billion years old, but we said also that we have evidences that the first living objects, simple living objects, for example, bacteria, appeared somehow 4.2 billion years ago. Sorry, 4.2 billion years ago. There are several theories trying to understand how this might happen. Probably one of the most interesting or one of the most easy to justify is that of Varela's Mces. Varela's hypothesized that in the what we call primeval soup that appeared on our planet, we have 4.2 billion years ago, we have very shallow oceans. Our planet is mostly covered by shallow water bodies, shallow oceans, in which there was a mix-up of all chemical substances, but let's say basic chemical substances, simple like nitrogen, phosphorus, silicates, and so on. But these chemical substances started to make or to assemble into more complex biochemical structures. And the first thing that should become apparent is that there should be a differentiation in this soup of molecules. This differentiation may be induced by, for example, radiation. Speaking with Schrödinger, that may be X radiation or any cosmic ray. So very energetic radiation arriving on the planet that somehow induced the formation of differentiated structure. And in particular, this differentiated structure should build some kind of borders. The idea is that in the primeval soup of all these elements, there is a differentiation in the structures, some more complex biochemical structures, let's say proteins, that may start building some kind of borders, cell that are the embryo, we may say as the embryo of cellular membranes. So the idea of Varela is that starting 4.2 billion years ago, due to cosmic irradiation, cosmic rays, there may be the appearance of some kind of differentiation in the structure, and this differentiation is linked to the appearance of, for example, borders in which we may identify an inside and an outside. The inside tends to be more stable due to the fact that there is a border, while the outside should be more perturbed due to the fact that it is more exposed to cosmic radiation. I recall you that cosmic radiation we are thinking at is very energetic radiation. So if you put together the Penrose law of the square root of n, the fact that we have the buildup of order from disorder structure. So in very disordered condition, there might be or might appear some thermodynamic constraints that drive to a differentiation or to an information, to a storage of information in some kind of structure, and linked to a order from order instance in which inside this structure the DNA, that is a proteic biochemical molecule, is a place in which information is stored. Putting all this together, we may understand how the first, or we may, if you want, if not understand, at least intuit, being intuitive, we may have a glimpse on how living objects shall be appearing. So, for staying simple, we shall recall that the first living things that we may talk about or we may have trace of are bacteria. I already anticipated that there are two main classifications of bacteria. Very simple one, prokaryotes bacteria on the left, and more sophisticated one, eukaryotes bacteria on the right. Of course, prokaryotes bacteria were the oldest one. Prokaryotes means, karyotes means nucleus, so before nucleus and with the true nucleus. The differentiation between prokaryotes and eukaryotes bacteria is the presence of a nuclei. Eukaryote bacteria contain a nuclei, and the nuclei is where the DNA is found. So what happens, or we may hypothesize that after the construction of these structures, borders, then we have an inside in which there are controlled conditions. So an inside in which the proteic assemblages that were constructed by the very large number of events with square root of n due to cosmic radiation. So the proteic assemblages were protected inside the nucleus. So after building the membrane inside, you have a kind of protected environment. And then in the subsequent evolutions, it took probably I recall you that bacteria dominated our planet for 2 billion of years, starting from the 4.2 billion. So there were only bacteria. And by a series of evolutive processes, and probably the most relevant one at least was symbiosis, different bacteria, prokaryotic bacteria, each of these bacteria were able to develop some kind of biotechnology, like, for example, photosynthesis. So some bacteria start to be able to produce, to convert solar energy into organic molecules. Some other bacteria were able to convert oxygen into organic molecules, like, for example, carbohydrates, and so on. So this is the invention of respiration. Then by symbiosis, a bacteria that is able of doing photosynthesis or respiration may be included in a larger bacteria, and this is the idea of how eukaryotes, so more evoluted microorganism appears. So we may say that eukaryotes acquired some of organelles by symbiosis, and these organelles may be found in the very complex structure of eukaryotic bacteria. Eukaryotic bacteria, as well as prokaryotes, have a border, a membrane protecting the inside from the outside. I recall you that according to Odum, one of the first definition of characteristic of a living system is a differentiation between an inside protected and outside unprotected, so outside chaotic, inside ordered and protected. So the appearance of membrane seems to be a key process in the evolution of life on our planet. Then we may make a step further, and we recognize that according to order from order instance of Schrödinger, and according to the discovery of Watson and Crick of the DNA in '54, we found, or we understood, that especially while in prokaryotes bacteria, the nucleotides, so DNA, was mixed into the cytoplasmatic liquid of the cytoplasmatic solution. In more sophisticated eukaryote cells, the nucleotides are found mainly in the nuclei of the cell, in which we have the very well-known molecule of ADN, the DNA. There are several types of DNA, of course, in the cells. In particular, we should distinguish between the nuclear DNA, that is here, and what is called the mitochondrial DNA. Mitochondria are, if you want, organelles that may be seen as the energy generators inside the cells. So mitochondria, for example, are those that are able to do photosynthesis, or simply to convert organic. Not photosynthesis, but are those that are responsible for respiration or the conversion of organic molecules into energy, ATP. So this process happens into the mitochondria, and there are some specific strains or filaments of DNA that is called the mitochondrial DNA, that is associated to the female, at least in mammals, to the female developing line. While in all species of eukaryotes bacteria, you will find the most part of information concerning the building of the structure of the organisms into the DNA. So now we may shall to start asking us if life is linked to the concept of intelligence. Intelligence, intelligere from Latin, intelligere means linking, linkage between different hierarchical levels. So intelligence has to do for sure with information management. So the management of information between different hierarchical levels. And we may ask ourselves, sorry, because I cannot read it anymore. Mm-hmm. 1 minute. Yes, you might agree with me that information management has to do with knowledge. So where are now? We discovered that living things build borders. They separate an internal environment that is stable they separate an internal environment from an external environment that is unstable and chaotic. The external environment is subjected to cosmic radiation, in which you have a square root of n number of events, producing a large number of different combination of elementary elements, basic elements like nitrogen, phosphorus, and so on, and producing quite a complex, starting to increase the complexity by producing more complex molecules like proteins, like DNA. DNA is protein. So we link our understanding of information management with an intelligent process. Intelligent process has to do with knowledge. So we pass we make the history of scientific thinking development in pills, 2 centuries in pills. We start from creationism, in which we thought that there should have been a divinity that creates the organism as we are looking at them. Then we see that thanks to Darwin, we have a switch in thinking toward evolutionism. Evolutionism means natural selection, so the changement in structure and in DNA due to external perturbation or cosmic radiation or whatever induced mutations. Mutations are saved, only those mutations that by natural selection seems to be positive for the new living objects. And then we arrive to the 50s of last centuries, 20th centuries, in which DNA was discovered. And as you understand, DNA gave rise to a dramatic new scientific approach, that is that of molecular biology. We may call it today genetics. So the dream was every information concerning a living object is stored into DNA. So this is the dream of biologists, molecular biologists, what is called genetics. Today, we are even a little bit further, and we are in what it is called the neo-Darwinism paradigm, but it might seem as the modern synthesis. So asking if intelligence or information management, or if intelligence is life, may be synthetically converted into the question, is DNA the representative of biologic intelligence? Can we trace down all intelligent or knowledge management processes to the existence of DNA? All the information related to the building of a new organism, is it included into DNA? Or we may say, are we intelligent because we think by gonads? Gonads, from a biological point of view, means are those cells, the primary cells, like, for example, spermatozoos or the ovules, the mammals are gonads, so are the cells in which there is the first appearance of the first recombination of DNA, so the first appearance of a new strain of DNA linked to the new organism. So, is intelligence codified into gonads? Are we thinking by gonads? Why are we posing these questions? Because the dream of the molecular biologists, and also the dream of big pharma, so pharmaceutical industries, was that of identifying one gene and with one protein. Let's say that one gene, genome is made up of genes. So pieces of genomes are genes. The idea is of molecular biologists, as we understand DNA today, is that one gene should be able to code for one protein. So one gene, one protein. We know that living things more or less display something like 100,000 proteins. So we expect to find at least 100,000 genes, or we would like to find 100,000 genes. Dramatically, it was discovered by the Genome Project, led by Dubeko in the 90s of last century, 20th centuries, that the number of genes are much more limited. For example, in a fly, we may find 25,000. In a worm, we may find 10,000. In a bacteria, 5,000 genes. So we are far away from 100,000 genes. What about the genome? Human genome has only 25,000 genes, like the fly. So it is evident by this experiment, by the Genome Project, that genetic determinism does not exist. So it is not possible to identify one gene to one protein. So the mechanism of protein synthesis is much more complex than we would expect or we would like it to be. So let us make a step back, how things appear to be, looking at how the modern biology developed. We have the evolutionism. Darwin, you may recall the very famous contraposition between Darwin and Lamarck. Of course, this was not a true contraposition, because Lamarck was never saying what he was thought or he was said to say. So, for example, if you cut the arms of a living animal, then its offsprings should be born without arms. This is simply stupid. was not the point of Lamarck. The key idea is that of a survival of the fittest. So survival of the fittest has to do with the effects of the surrounding environment onto living objects. So the neo-Darwinism tells us that only those mutation that give to the offspring, to the new organism, more possibility to survive, will be transmitted. We may talk of inheritance of acquired traits or characteristics. So we start to talk of the possibility of new organisms to transmit to the offsprings the new acquired traits. The interaction of environment or the external system with the living object today is called by biologists epigenetic. Today it is clear that even if living organisms appear to have a fixed quantity of coded information to the DNA, the possibility of converting or the process of converting this coded information is dramatically driven or influenced by the surrounding environment. Epigenetic is the study of changes in phenotype. You know, genotype is the quantity of information contained into the genes. This information may be, let's say, manifested through the phenotype. The translation of information of genotype to the phenotype is dramatically linked to the external environment. Now you understand that from our perspective, the story is much more complex than that of genetic determinism. Epigenetic may be seen as any activity of gene regulation through chemical processes but do not involve changes in the genotype, but may modify the phenotype of the individual and or the offspring. So genotype is the study of gene expression. Sorry, epigenetics is the study of gene expression. Gene expression control, as we said, is made up or is dramatically influenced by the external environment. Biologists know very well that during morphogenesis, so the changement of the expression of genes, we have totipotent stem cells, the cellule staminali, but totipotent meaning they could be converted in every cell of a complex body, becomes the various pluripotent cell lines, for example, the cell lines linked to the construction of organs, like for example, liver or skin or brain or eye cell lines. So during morphogenesis, totipotent stem lines become various pluripotent cell lines of the embryo, which in turn become the fully differentiated cells. So when an embryo starts or appears, we have that, for example, let's see the example of mammals. Mammals, we have that, for example, the ovuli, that we may represent like a round, is impacted by a spermatozoa. So these ovuli and the spermatozoa are single cells. When the ovule is impacted by the spermatozoos, in the point of insertion, there is a breaking of symmetry in which the ovule is subdivided in 2 points, what it is on the left and what it is on the right, in respect to where the spermatozoa entered into the ovule. And now we have the first differentiation of the cell. So in this process, the ovule gonadic cell is impacted by the spermatozoa, which establishes a first line of symmetry or breaking of symmetry in the ovule. And it is exactly along this breaking of symmetry on this the insertion line of the spermatozoa that the subdivision of the first cell appears. So cellular division starts happening here. What it is going from here, when we end up with 2 cells. These cells are not gonadic cells anymore. Instead, these cells come from the subdivision of the first mammal cell, the first ovule cell. The subdivision is linked to the insertion of the spermatozoa. So here we have 2 cells that are identical because of the subdivision. They are sharing the genetic information because the mixing of the genetic information happen exactly in this point when the spermatozoa enter into the ovule. So we have the appearance of 2 new cells which share not only the genetic code, but also shares the structure of the membrane. You see, here you have the first mammal ovule cell. When the cell subdivides, the new structure contains the same genetic code, but the membranes of the new cells are shared by the new cells. At this point, it becomes evident that the control during this differentiation or the control of gene expression is somehow linked with the external environment, the perturbations coming from the surrounding environment. Okay, we may also say that the perturbation may also be linked to what we may call growth noise. So this accounts for some level of stochasticity in the process. But the important point here is that for sure the external environment is somehow influencing gene expression control. So now let's give a look more closely to what is the cellular membrane. Let's give a look on how membranes may somehow be linked to evolution. A cellular membrane is a lipophilic saccaric layer, lipid layer, with some holes. You may imagine this as a lipid layer with some tubes that allow connection from external to the internal environment. So the proteins, so complex biological cells, interacting from the outside with the inside of a cell, are transferred from outside to the inside through some kind of tubes or pathways that are specific, very specific for the particular proteins. So this structure is very elaborated. What we may recall, the most similar object we may look at, it is a semiconductor. You may recall that a semiconductor is a wafer or a layer of silicate in which some kind of drogens are inserted, gallium, germanium, and so on. And are exactly these drogens that gave the very particular properties of the semiconductors. It is strikingly apparent that what we most think to be close to intelligence is computers. So, but let's say that the most close to intelligence artifacts made by man are computers. All computers are based on semiconductors technology. So somehow the interaction from an outside with an inside has to somehow be linked to the evolution of a cell. So we may pose again our question, where is intelligence now? Of course, intelligence is not in the DNA. Why is there is another important experiment I should talk about, made by biologists immediately after the discovery of DNA. They took eukaryotic cells. They start studying bacteria's eukaryotic cells, and then they enucleated these cells in order to see what happened. And what happens to an eukaryotic cells if you take away the nuclei? So you take away where intelligence we thought is in, so genetic code. It happens nothing but that the cell cannot replicate anymore. So what it happens into an enucleated cell, the enucleated cell continues performing its living tasks unless it dies up because of disruption of a structure, and the enucleated cell is not anymore capable of replicating or repairing itself because it has lost the code needed for building the particular embedded structure. The surprising thing is that if you enucleate a living cell, the living cell continue to be living. So where is the intelligence? It's not in the nuclei, it's not in the DNA, but we may say that intelligence is somehow linked to the model applied for the interpretation of reality. Living systems happens to respond to external stimuli in non-trivial ways. From the model point of view, we may say that living things have a model of interpretation of reality that is non-trivial. So the cognitive model for the interpretation of the reality may be seen as the place where intelligence stands. Did I convince you? I hope so, because I'm quite convinced of that. So if you ask me, what are living organisms? Living organisms are organisms that are able to make a cognitive model for the interpretation of their reality. A cognitive model, I'm not specifying anything about a cognitive model. The only thing I'm saying is that a living object reacts in non-trivial way to external stimuli. So we may make this play of words, and we may say membrane Somehow intelligence may be located into the membrane of a cell. So at this point, we go to the finishing of this lecture by making a small a small reflection concerning signaling. We said that evolution has to do with gene expression control. Gene expression control is linked to external stimuli. We said that external stimuli is mediated by the very general model of interpretation of a reality that in the basic elementary organisms like the bacteria may be identified with the cell membrane. Then we said that the cell membrane is a layer of lipids in which there are some kind of canals embedded that allow the passage of chemical molecules, proteins, or chemical signaling. So if we analyze the speed of chemical signaling, we find out that chemical signaling speeds are on the order of centimeters per second, while electromagnetic signaling is in the order of the speed of light, so 10 to the 5 kilometers per second. My question is, if you are an organism of various trillions of cells, like complex organism like a mammals is, and you would choose a mechanism of signaling in order to coordinate the behavior of these trillions and trillions of cells, which signaling method would you employ? Chemical or electromagnetic? Of course, the answer is trillions and trillions of cells. Probably the most efficient is electromagnetic signal. This is an argument to say that we cannot neglect the effect of electromagnetic signaling on the behavior of life. So when we ask ourselves if electromagnetic waves or electromagnetic signals are somehow interfering with the development of biological processes or bioprocesses, the answer is it is very likely that electromagnetic signals somehow are involved at a different layer, at a different hierarchical level, if you want, because chemical signals are much more stronger and much more specific, but they are much more slower than electromagnetic signals. But we cannot, in principle, neglect the effect of electromagnetic signaling on living systems. So this opens a new strange of thinking, biology and quantum mechanics. There is a materialistic prejudgment or prejudice, or the so-called mechanicistic physician, Newtonian physicist, uses hormones, cytokines, growth factors, suppression, and so on to describe the behavior of living objects. So new biologists or modern biologists like the mechanicistic paradigms, but inherited by physicists. Physicists had to abandon the mechanicistic paradigm with the invention of quantum mechanics, in which one of the fundamental postulates is the Heisenberg principle, the indetermination principle, Heisenberg principle, in which the physicist is forced to admit his inability to know with arbitrary precision the behavior of a system. And then we ask to the biologists and the role of electromagnetic fields is simply to be neglected? I leave these questions open to you because it is, I mean, I do not have an answer, but I have a striking suggestion that electromagnetic fields shall have an important interference in evolutionary mechanisms, so in living processes. Okay, for now, we stop here today. If you have any questions, I wait for your question for 10 minutes. Otherwise, I close the channels and I go to drink a coffee.